So welcome on my behalf. Today we're talking about this single-cell RNA-seq data analysis using Chipster online learning course. My name is Maria Lehtivaara. I work here at CSC as a bioscience support specialist, and I work with the Chipster software. And here with me I have my colleague Eija Korpelainen.
Hi.
So Eija is working in similar tasks and is in charge of Chipster. So we will both present today. Just a second, I have to figure out how to change the slide. Okay, here we go. So the topics today will be, well, we present this new massive online learning course for you. We will give you a short overview of the single-cell RNA-seq analysis and a brief overview of Chipster software, which is used in the course. And then we check that you can get access to both this course and to Chipster and know where to get assistance when needed. Brief motivation. Well, the course is, it doesn't have much prerequisites. It should work for everyone since we are using the Chipster software, which is easy to use and you don't need to know R or any other language. So it's really for everyone, and it's really good to know how to do your analysis, even if you have someone else to do it for you. Knowing the steps and the different tools helps you to discuss the data analysis steps. And usually the one who designs the experiments knows best about the potential batch effects and the biology involved and overall about the experiment. And when you know how to do the data analysis in the end of your experiment, you also know how to design a good experiment and you're not wasting money on poorly designed setups. However, if you are a bioinformatician doing the analysis for someone, it's also good for you since you can easily learn in this basic level course, and you learn how to do the analysis using Chipster, but then it's easy to move to R if you decide to use for example R to do the proper analysis. Right. So about the massive online learning course that we have developed in our e-learning platform, which we call eLearning or e-Lena. Here's the address, and you can log in with your Haka account or Elixir account and then self-register to the course. In the course we have video lectures and some exercises which you will be doing with Chipster software and small quizzes. And in the end you have this test your skills section where you analyze your own data more independently. And there's also this questions and answers forum for discussion. So there you can get help and see what other people are struggling maybe with. So you can share information nicely there. After you have completed all the exercises, you can download a course certificate, just like in our normal CSC's on-site courses. And we recommend 1 credit for passing this course. The course is free of charge and it's open now and until the end of this year. We will take a closer look in the end of the presentation, but this is what it briefly looks like. So here you can see the contents of the course and here's this completion progress bar. I'm sorry, I'm moving around these 2 objects here, so sorry. Yeah, and then here you can see the different sections that we have there. So you can see that we have 10 different topics or sections, and under each there's a video lecture, brief explanation on what that chapter is about, then some exercises and the quizzes. Yes, and we will be using this Chipster tool that looks something like this. So Eija will give a introduction in a couple of minutes about this software. And like mentioned, you can log into Chipster using your Haka or Virtu credentials, or if you already have a CSC user account, that also works. If you don't have either, you can always email us and ask for a training account. And Chipster runs on top of your browser, so you don't need anything more besides that. And like I said, you don't need to know any command line or R or Python language. And we feel that it's really easier to learn using this graphical interface, even if you then end up using R later on. That being said, maybe some basic knowledge about sequencing and molecular biology might be handy. So basically, that means that you know a little bit like how the sequencing works and what is an RNA molecule and so forth. Yes. In the course, you will learn how to use the Surat tools that are available in Chipster to analyze the single-cell RNA-seq data. You can name and discuss the different steps involved in the analysis and understand the advantages and limitations of such analysis in general and in Chipster. There are basically two tutorials or pipelines that we are dealing with in the course. So the first one is the basic analysis, where you are finding the clusters or subpopulations of cells and their marker genes. And then in the second part or second analysis type, you are comparing two samples. In our example, they are treatment and control. And there you are identifying cell types that are present in both samples and getting the markers that are conserved in both samples, so cluster markers basically. And then you compare the samples to find some cell type specific responses to the treatment. And of course, you will learn how to operate the Chipster software, which can be used to other things as well. So that was the introduction to the course. And now I will try to give a very short introduction to single-cell RNA-seq data analysis. So it's a relatively new technology. And that means that the methods are actively being developed. There's new versions of tools coming very often still. And if you compare to the traditional bulk RNA-seq method, so there you take a sample which has many cells in it, and you sequence the whole thing as one. So you get this average gene expression profile. So it's average from all of these cells. So you don't really see the cellular heterogeneity. Whereas with the single cell RNA-seq, you take one cell at a time, or you label them and sort of sequence them individually. So each cell type has a different unique expression profile based on which you can cluster the cells and find these subpopulations based on the expression. So you can find out many more things with a single cell RNA-seq that you cannot with the bulk RNA version. There are many technologies available for single cell sequencing. There are droplet-based, plate-based, and well-based technologies, out of which these droplet-based seem to be the most used ones. And this 10x Chromium and Dropseek are maybe the most used ones. And these are the ones that we are talking about during the course. One thing to mention about single-cell RNA-seq methods is that the libraries are usually 3' tagged, which means that only the short sequence on the 3' end of the mRNA is sequenced. So that causes some limitations. Sorry. Yeah, to go through a little bit more detail, what happens in the beginning of the analysis. So this is from Dropseek, these images. The paper is mentioned here. So basically in Dropseek, you aim to get this bead, one bead and one cell in one droplet. And these beads consist of these yellow cell markers. Oh, sorry, and that's same for the whole bead. And for each of these tails here, there will be a unique molecular identifier, or UMI, and then a long poly-T tail, which will attach to these mRNA molecules. So when you sequence this, so each cell will attach to one of these beads, or the molecules from that cell. And then when you sequence the whole thing, all the cells within these beads, you get this kind of data. So for each read, you get a cell barcode, the unique molecular identifier, and then on the other read attached to this one, you get the actual RNA sequence. And this kind of reads, you get hundreds of millions from your sample. From this, so this is usually in the FASTQ format. So after this, after you got this this file, you align these sequences to genome, to reference genome, and then group these reads based on the cell identifier. So here you can see that the reads with the same red sequence correspond to this cell 1. And then you can see, based on the alignment, that which genes they are aligning to. And then you can count how many reads or how many molecules were found in this cell for this gene, and form this kind of table. Now the UMI comes handy here, since here you can see that these two reads are mapping to this same gene. And if you look very carefully, you can see that the UMI sequence here is exactly the same. So now you know that they are actually from the same original mRNA molecule, and they are actually PCR duplicates. Whereas here, they have the different identifier. So these are counted as 2. So they are originally from 2 different mRNA molecules from the same gene. So that's where the UMI is used for. So as an end result, you get this digital expression matrix where you have the genes here and the cells here. So if you remember if you have been analyzing normal, like bulk RNA-seq, there you have a similar table where you have the genes here, and then here you have the samples. So let's say 6 samples. So now instead of 6 samples, you have thousands of cells. So it will be a big table. Now, this first phase, which I described here, so starting from the raw reads and ending in this digital expression matrix, this is not covered in the course. However, these tools for this kind of Dropseek data are available in Chipster, and we do have some tutorial videos on how to use those tools as well. But since the analysis often begins from this matrix or from similar output from the 10x device, that is where the course actually begins. So in the first part of the course, you will start with this digital expression matrix, and you will end up with these clusters, so sub-cell populations and their marker genes. And these would be the steps involved in the first step, or the basic single-cell RNA-seq data analysis. The second analysis type that will be covered in the course is this 2-sample analysis. So here you started with 1 digital expression matrix, and now you will start with 2. So you have 2 digital expression matrix as input, and as output you will get clusters for the 2 different samples in your data, and their conserved marker genes and the differentially expressed genes between the samples. So in short, this is possible to do in Chipster, but usually don't have to do this. So usually you start from this digital expression matrix, and this is the these are the 2 analysis types that will be covered in the course. To go through in a bit more detail the different steps involved in the first type of analysis, we start by checking the quality of the cells. So you remember that you had thousands of cells in your data and tens of thousands of genes. So you start by filtering out some low quality cells. So you will learn how to identify the low quality cells and how to filter them. Then you do some normalization for the expression values and identify highly variable genes, which are then used later on for the principal component analysis. Between those two steps, you also scale the data and regress out some unwanted variation, so technical variation that you don't want to see when analyzing the data. You learn how to find these highly variable genes. There will be usually 2,000 of them out of the tens of thousands of genes in the original data. And then you use this principal component analysis to that is performed on top of those 2,000 variable genes. And this is a dimension reduction method. And after this, you will only have, let's say, 10 different dimensions, which you use to cluster the cells. So you're reducing the amount of data, but maintaining the variability in the data to do the clustering. After you have the clusters, you can visualize them using these TSNE or UMAP methods. And after that, you can use this statistical testing to find the the cluster marker genes, which you can use to recognize which cells belong to which cluster. So there are many quite different steps compared to the bulk RNA-seq. And you will learn a lot about different dimension reduction methods and about the clustering, especially in this type of analysis. So all these steps and topics are covered in the small sections of the course. So each will have their own short lecture video demonstrating these things, and then you get to exercise those using Chipster in the exercises. In the second type of analysis, we are integrating two samples. So whereas here we only have one sample with many cells in it, here we actually have two samples with different cells in them. And in our case, they are the control and treated sample, but they can be any two samples that you want to compare. And this is, of course, the ideas that you learn can be broadened to a different setup also. So there you identify the cell types that are present in both samples and obtain the cell type markers for those cell types, and then also find cell type specific responses to the treatment. The trick there is that we need to find the corresponding cells in the two samples. So we want to get rid of this patch effect coming from technical and biological variability. So if you compare these steps of the second type of analysis to the original basic analysis, there are some differences. The beginning is very much the same, so you do these things for the both samples in your data, but then you integrate them. And for that, you need this canonical correlation analysis. So it's yet another dimension reduction method, if you want to put it that way. And that is used to align these samples so that you get rid of the batch effect. Then again, familiar scaling and PCA happens, the clustering, visualizing the clusters with these methods that you learned in the basic analysis. And then again, something a little bit different when you're doing the statistical testing. So now you're also, well, you're looking for the biomarkers for the clusters, but also looking for the differentially expressed genes between the samples within the clusters. And of course, the visualization is a little bit different here. So you learn about that as well. Important thing to mention. So in the exercises, we are using these Chipster tools, which pretty much follow these steps mentioned here. However, we haven't developed the tools within. So we have developed Chipster, and the tools within Chipster are then originally from this Surat R package, which is one of the most popular R packages used nowadays for single-cell RNA-seq data analysis. So if you want to, you could follow the same exercises using the Surat tutorials, because we are following very carefully 2 of the tutorials that they have in the Surat web page, which you can see the address here. And similarly, we store the data in these Surat objects. So it's a massive object that contains all the information in one big block. And in Chipster, you can move these R objects back and forth. So you can import them from R to Chipster, and again from Chipster to R, if you want to move between these 2 platforms. Surat tools are not the only tools that we have for single-cell RNA-seq data analysis in Chipster. We also have these scatter tools that can be used for quality control as well. And then we have these SC transform tools for normalization as well. Okay, so that was what I had to say about in the very short introduction to single-cell RNA-seq data analysis. Now we could take a look at Chipster software, which is used in the exercises. So maybe I stop sharing, and Eija, do you want to continue from here?
Okay, so let's move on to the software that is used in the exercises of this e-learning course. So some of you might have used Chipster already, but so basically it's a graphical software that we have developed at CSC for our users in the Finnish universities. And the idea is that through Chipster, you can easily run analysis programs that you would otherwise be running on command line or using R scripts. But when you use them in Chipster, you can operate them with mouse. So there are quite a lot of analysis tools. Single-cell RNA-seq is just one area, but there are many others. And Chipster is free and open source. So you can analyze data, visualize it, and also share your analysis sessions. I will be showing that in the demo. So the Chipster website currently looks like this. In the course, we are using the Chipster version 4, which is a web-based application. You still have the link to the Java-based one as well, but that will be removed soon. So this is the one you go for. And otherwise, from this front page, you will always find information about upcoming training events. And there is also a link to our YouTube site with a lot of tutorial videos. And the other link I might want to point out is the course material link here. So this is a script, Chipster screenshot. I will be showing all these things to you live shortly. But just to point out that there are various sections in this interface. So you have the files view here as a list of file names, or this kind of workflow view where the files are shown as boxes connected with arrows, just to help you to remember what file came from what. So every time you run analysis, it creates a new arrow and new result files. Then the analysis tools live here. They're under 3 different tabs, and under each tab there are several categories. So for our MOOC course, you will be using the tools in the single-cell RNA-seq category. So once you click on the category, you will see the tools underneath. And then you can, so if you have selected your input file, the category and the tool, you can either hit run or fiddle with the parameters and click run. And when you start the job, you will have this little thing moving here, indicating that the analysis is going on. And finally, down here, when you select the file, it will show you the contents here and different visualizations for it. I also want to mention that there is a built-in support functionality in Chipster. So if you get into trouble, if you get an error message, for example, rather than emailing us and copying the error message, you can just use this contact support function, which you will find in the upper panel. So here, the last thing here is contact. And when you click there, you will get this page where you have the contact support button. So here you type your message, the question to us, your email address, and then you have the option to allow us to look at your data. So if you click here, you give us the right to look at your sessions, so then we will see exactly what you have done, which often makes it easier to troubleshoot. And if there was an error message, that would be then automatically copied to the message that we get through this system. For more information about Chipster, you can always email us, so chipster@csc.fi. There's the YouTube channel, as I mentioned, and also a page with all our course materials. So let's go to Chipster itself. So this is when it looks like when you get there. So the first thing you need to do is to log in. Here you have the different options that Maria was mentioning. So you can log in with Haka or Virtu, or you can have a CSC account. Now in my case, I used a demo account here. And so this is what Chipster looks like when you start. So now it remembers that I was actually working on this session before. But if you didn't have anything to start with, the first thing you would do is to add a file. Now, in your case, for the course, what you need to do is you need to go to the sessions. And down here, when you scroll down the list of sessions, you can see that, well, there are your own sessions, but also example sessions. So we have quite a lot of different analysis examples here. And when you scroll further down, you see that there are training sessions. So in the training sessions, there is a session called Course Single-Cell RNA-seq Seurat, which is the starting material for your session. All this is in the instructions in the e-learning platform. You can also see that there's another session with a very similar name, except that it says done in the end. So this is actually a ready-made session which contains all the analysis steps. So if you get in trouble when doing the exercises, you can also always go here and check and compare if something looks different from your session to figure out what went wrong. And like Maria said, the course covers two types of analysis. So there is one sample analysis where we find clusters of cells in one sample and marker genes for them. So that's this one. And then there is integrated analysis of two samples. And there is also the done session, the model session for that. But so now let's start with, like you would be starting with this session. So here is your input file. This is a tar package of the 10x files. And so you would select that as input. Then under NGS tab, you go to the single cell category. And then here you have the available tools. So like Maria said, many of them are based on the Seurat package. And currently we have Seurat version 3, which we will update to Seurat version 4 once it becomes stable. It's still a beta version. So there are tools for various things. When you scroll further down, there are some tools for pre-processing of Dropseek data. Maria also mentioned this in the slides. So in the course, we don't do this part at all. We just focus on starting from the digital expression matrix and then take the steps toward clustering. So anyhow, so now I have selected my input file, my category, my tool. And here I can see a little text explaining what this tool does. If I want to know more, I can click this more info here, and it will take me to the manual page of that tool. If some of you are more bioinformatics oriented, you might also want to click on this tab here, which says source code. So that actually shows the code that the tool is running behind the scenes. Now, if I just clicked here, it would run the tool with the default parameters, but often it's necessary to go and check and potentially change the parameters, so that you would do by clicking on the parameters button. This particular tool is very simple. There are only 3 parameters. But what is important to check is what is underneath here. It says input files. So this particular tool can take 2 types of input files. My type here is a 10x output files in a tar package, so hence I have to check that it was assigned correctly to here. But anyway, now, so let's say that we wanted to change the project name. So these cells are peripheral blood mononuclear cells, hence I give this name. And now I can click run. When it starts running, you see it here. And if you click on the jobs, it will tell that this particular job is running. This is a very quick job. So you can see that the result files came already. But sometimes if you have a longer thing and you wonder what is going on, where it is going, you can click on that name of the job and it will show you the screen output. But anyhow, so here now I got 2 result files. So there is the R object that Maria mentioned already. And like she pointed out, it's always this R object which actually contains the data. So the PDFs are just plots, but the R object is what we always give as input to the next tool. So for example here, the next thing would be this one, filtering cells, normalizing, regressing out unwanted sources of variation, and detecting variable genes. So I would do like this. Again, in this case, there are actually lots of parameters. And then I would click run. So while that is running, let's look at the other output file, which is the one you are meant to look at, so the PDF. So if you just click there, it will open it here. Sometimes this area might not be enough for viewing what you want to view, so you can always click open in new tab, and it will open it in your browser in a new tab. So in this particular case, for example, we have quality control plots of cells, and all these plots are, the content is explained in the videos in the e-learning course. And down here you can also see how many cells there were. Okay, so meanwhile, while we were looking at that one, the next job actually completed. And as you can see, again, it created a new R object. So this will happen pretty much every time you run a tool. And every time, so you start with one R object. When you run a tool, it adds new things in that object. And in Chipster, it in practice creates a new object. So when you do the exercises, it's important that you always select the latest R object for the input of the next tool. Okay, so then let's go to the other, maybe one more thing. So if ever you wanted to change names of these files, that would happen here. So now I have this PDF selected. So if I go to these 3 dots here, it will allow me to rename it, delete it, or export it, meaning that I can just take that particular file out of Chipster. Now, if I just click somewhere else so that no file is selected, then in the same field here, I actually have information about this whole analysis session. And again, if I click here, now I get different options. So I can rename the whole sessions or edit notes. So notes means basically the text you write for yourself to remember what is going on. And the other important thing here is that, well, you can download the session to your laptop, or you can make a copy, or you can share the session with someone else. So let's have a quick look at that. So the sharing window looks like this. So you need to, so basically you go to add rule and then you decide with whom you want to share the session. And here you need to write the Chipster user ID of that person. So for example, if you wanted to share it with me, you would write here, if you were using my Haka ID, you would be writing like this. And then you would need to decide how you share it. Do you give me full access to your session, which means that I can also delete things from there? Or you might rather want to be careful and just say you want to share it in a read-only fashion, meaning that I can just look at it. So then you would save this rule, and I would get a message in Chipster saying that you have shared your session. So this is a way how you share sessions with other Chipster users. All you need to know is their user ID in Chipster. And if they don't know what their user ID is, you can tell them to go up here. So here it says account, and here I can see that now I'm logged in as user called demo, and my user ID is now this Yas/demo. Okay, so that's about it. Maybe one more thing. So of course these files here, you can move them around the way you like. You can, if you need to select several files, you can draw a box, which is easier than clicking many of them. And here in the list, you can actually see the file names, so you can also select files here. And if you right-click, it allows you to delete as well. So now to finish with, I just want to quickly show you the other session. So now I was using a course session, so it's asking me if I want to, my own copy of it. So no, I don't at this point, but I just wanted to show you this other session. So this is now the integrated analysis of 2 samples. And maybe just to show some plots from here. So we have, as we do the integration, we can see this kind of alignment plots, for example, how well the 2 samples, blue and red, go on top of each other, how well we manage to get rid of the patch effect, things like that. Here I have my list of differentially expressed genes in a particular cluster or conserved market genes. And here I have various visualizations which can be handy. And again, if this area is too small for you, you can open them in a new tab. So I think I have actually opened this here. So there are quite nice visualizations of different types. And again, all these visualizations are explained in the tutorial videos. So I will stop here.
All right. So let's check quickly what the course looks like. So if you go to e-learn.csc.fi, the page will look like this. And you can see here that you're not logged in. You can still see all the courses we have available at the moment. And here you can log in. So I'm going to use my Haka account. Right. And then you can go to this Singlecell RNAseq data analysis course. So what you should see is this completion progress bar here. If you don't see that yet, you haven't enrolled yet to the course. So to do that, click here. Oh, I can't demonstrate it now with this a bit special setup. I can show it from the slides. So here, you should see this cogwheel that I was trying to demonstrate a moment ago. And if you click that, there should be this enroll me in this course option. So then when you click that open, something like this opens up, and then there's this self-enrollment section where is this enrollment key bar, and there you type this key, which for this course is SC RNA-seq Chipster, and then you just click enroll me, and then after you have done this enrollment, you should see this completion progress bar. Right. If I go back to eLearning, now you can see the bar here, so that's good. And here you can easily follow which exercises you have already done. So green means it's done, and blue means it's not yet completed. So here you can see, for example, the Chipster quick tour was done, and course feedback was completed. Of course, you want to complete that in the end, but yeah, you get the idea. Here you have a brief about section of the course, so course contents, learning objectives, links to relevant materials. So here you can always find the course slides and exercises and other materials, which are available for you also after the course. So this course is now valid until the end of 2020. There's instructions how to get access to Chipster, a link to Surat tools, or the guided analysis there, and an article about sample integration in Surat, so a little extra material there as well. Here are some practicalities. And then this is quite nice. So this is the question and answers forum. So we really hope that if you have any questions during the course, don't be shy to type them here. If you put them here, others can also see them. So it's very, very nice for sharing information. And you can also answer if someone is asking something that you already know the answer for, please go ahead and answer. Then here you can see the different topics, which are also available here. And you can just click them open, and you can see that there's a short description for this particular section. Then usually there's a tutorial video, which you can open here. It starts playing there, and you can put it also on full screen if you like. And with ESC, you can escape. Great. Or you should be able to open it in YouTube as well. Okay, then we have usually some exercises that you are supposed to do using Chipster software. So they are quite detailed, and you should be able to follow easily, but if you don't, remember the Q&A forum here. Then we have some exercises or quizzes or something similar here. And here you can now see that once this is completed, so once you have answered the questions or done the tour in this case, this tiny little box here gets ticked. So that tells you that this section is completed, and you can also follow the same here in the completion progress bar. Right, so then you go through all the sections. So here in the beginning, we do the one sample analysis, and then later on we do the comparing of two samples. And then the section number 10 is testing your skills. So this is something that people hoped from us. So there you get to try on your own data, or if you don't have your own data, you can download from web. There are a lot of data sets available online. Or if you don't want to do that, there's also this Dropseek done session from which you can continue and do the one sample analysis again with the different data sets. So these are examples of what you can do in this section. So the idea here is that because always in the courses, the demo data sets and the demo sessions are pretty straightforward because we want them to go smoothly and people to learn about the different steps. But that's not very realistic in the sense that usually you will run into some difficulties when performing the analysis for the first time, or the second time, or the third time. Most often there's something, especially in the data input phase. So usually you have to tune the data somehow. So that's why our course participants from the previous courses have wished for this kind of section, and here we have it now. Here are some instructions on what you could do and what you should maybe focus on. And then there's this final assignment where you can share what you have learned. So that's really from this section especially. So what did you find difficult? What were the new skills that you learned, and so forth. And again, if you run into difficulties, there's the Q&A forum, or you can email chipster@csc.fi, but we really recommend the forum. It helps others as well. Great, then you have completed the full course. There's the last section, which is called the course certificate and feedback. So we really hope that you give us feedback. Actually, we require you to. It's one of the tasks here. So there's a link to the feedback. And once you have filled that and completed all the exercises, you can download the certificate from here. So now you can see that it's enabled. So all the quizzes are not completed yet. So once they are, you should be able to download it here. Great. Yeah, I think that's what I had to say about this. Let's see. So let's see, do we have some questions here? Meanwhile, Eija, do we have something else we want to mention?
I cannot think of anything right now, except maybe just saying that really with any problem whatsoever, you can just email chipster@csc.fi and we will sort it out. And the other thing I didn't mention in my Chipster slides was that, well, as we are the developers of this software, we would be super happy if you send us comments about the software. So when you are using it, you might notice that something is not working the way how you would like it to work, or is a bit clumsy or whatever, or some analysis tool is missing, or whatever it is. So don't hesitate to email us. We don't get angry. We are just happy if you let us know. So this is how it has worked over more than 10 years now. We have got a lot of feedback from users, and that has been super useful. So it has really allowed us to shape the software so that it's most useful to you.
Excellent point. And we actually have a section for that in the course in eLearning, where you can comment these things. So it would be very nice if you could do that.
Now there's a question regarding, so is there any Chipster implementation for Python? Do you mean if there are any tools based on Python scripts in Chipster? If yes, I can say that yes, there are, but was that your question? Or like if you can integrate Python-based scripts in Chipster?
Yeah, so Chipster is, okay. So Chipster is, we are wrapping different tools from different developers inside Chipster. Some of them are in R, some are in Python, some are command line tools. So different things. And moving back and forth from, let's say, R and Chipster or Python and Chipster is of course possible by importing and exporting the data back and forth, if you like to do that. But yeah. Okay, now there's one question. I was thinking if I can, for example, control and execute a pipeline from a Python Jupyter notebook inside Chipster. No, you cannot do it that way. I think like so we need to implement the tools in Chipster. So the question there was in the chat was that how about if one does not have University Haka account, can somebody still register for the course and get certificate? Okay, so that's actually about the eLearning then. But yeah, for Chipster, if you don't have the credentials, we can create you Chipster credentials. Was it ASO that we agreed that you can get them for 3 weeks?
Yes, Yeah, correct.
Yeah,
So you can always email to chipster@csc.fi and we will sort it out. So for sure you will have credentials to do this course.
Yeah. Now it's 2 o'clock. I would like to still say very much thank you to all for participating. And here's one more. The instruction on how to contact us. So you can always email us. There's the forum in eLearning where you can ask questions, which is recommended because others can see them as well. And now you can start working with the course, and you have until the end of the year to do it. Yeah, that's all I had to say for now. Please don't be shy to ask us anything. And on my behalf, have a pleasant week and thank you for participating.
Yep, thank you from my part as well. And as you are working on the course, if you at some point realize that that sort of question and answer session might be useful after all, just let us know and we can schedule one.
Yeah, and we will let you know for in the training pages, so you can always find our upcoming webinars and trainings there at csc.fi. Thank you.